And so I want to say just before we jump in, I want this to be a conversation, so feel free to interrupt at any time. But I know that feels sort of awkward when, everybody else is quiet. So feel free to drop stuff in the chat if you feel more comfortable with that. All right, so let's get started. This is Epigenetics on AnVIL. It's part of a project that I'm working on with Ifeoma Nwigwe at Johns Hopkins. I'm at Fred Hutch. But she's been really instrumental in this. She's an epigenetics rock star. So we're trying to make just sort of the topic in general a little bit easier to understand, especially in the context of using it in the cloud or working on AnVIL. So we've called it nature versus nurture, try to really boil it down to kind of the theory there. But let's get into it a bit. All right. So first, we'll start with a little bit of background. I think most of us here are pretty familiar with just genomics or DNA in general, but maybe we're a little rusty on our epigenetics and thinking about the experimental approaches. So we'll review some of that. If you've already studied it before, it's always good to have a little bit of a refresher. And we'll get into a demo to look at some of the ChIP-Seq experimental results on AnVIL. And then we'll talk a little bit about, if you don't have your own results already, or if you're not using results from online somewhere, how would you start from scratch on AnVIL? Obviously, it's a little bit involved, so we won't get through everything today, but I'll kind of show how we could get started, as well as some other options for looking at epigenetic experiments and results. All right, so background first. Move around my windows a little bit, sorry. All right, so this Time magazine cover is a little bit dated at this point, but in 2010, I feel like this is a good milestone to think about. But in 2010, many people learned about the idea that DNA doesn't dictate our future. DNA isn't our destiny. And folks started learning about epigenetics in the general public and how scientists were discovering how the code itself is not the whole story. So I love this lead here, the new science of how epigenetics, how it reveals the choices you make and how it can change your genes and those of your kids. So huge impact. And I think, even though this is sort of the general public's introduction, I think this is when it really took off because, of course, you can't separate the science from what people are thinking about in everyday life. So epigenetic research really kind of exploded after this. And thinking about where nature and nurture intersect, and we want to understand kind of how those choices we make can affect our genes and, whether we make those choices or they happen to us, how all of that interacts. And I think that's become a lot more exciting and growing area of research since 2010. And as an aside, the epi prefix, I think it's always good to kind of think about where these words come from. So epi meaning on or upon, it comes from Greek. So if you think about things like epidermis, it's on your, skin. But epigenetics really thinking on top of the DNA itself. And so, how is that DNA sequence? How is that actually modified by something that is on top of it or around it? So along those lines, I just mentioned how it's the environment around you, what can happen to you, or the decisions you make. The idea of the exposome is a key concept in epigenetics. So no longer thinking about just the code itself and heredity and what your parents' genomes are, but the actions taken throughout your lifetime and external factors that influence your experience. So what we eat and do, where we live and work, but thinking about not just the genome, but the exposome as well is pretty fundamental to epigenetics. Okay, so a little bit of review. This is just, I think one of the coolest things about biology is that DNA gets packaged up into chromosomes. If you've ever taken your headphones out and you find that they're super, super tangled. If we didn't have a way to package up our DNA, that's what would happen, right? It would be a huge mess. Nothing would ever get translated or transcribed. And so we have this really remarkable phenomenon where DNA is packaged up into chromosomes, super coiled, even so much that we can actually visualize them with powerful microscopes. And to hopefully recap a little bit, but again, always nice to review, is that histone proteins, so not just the DNA at this point, but histone proteins are responsible for packing that DNA up into chromosomes. There are a number of different classes of histones, and each of these contain a tail that are rich in lysine and arginine. Okay, and so this makes histones positively charged. And if we think about our chemistry, positive and negative charges tend to attract. And so our histone proteins and our DNA like to come together. And so that's how that kind of at the fundamental level, we can get that DNA to behave and to wind up. And so it's a little bit more complicated than that, but those histones that I mentioned, they come in this unit, and they can have all kinds of modifications to determine how tightly packed the DNA is going to be. So in the case of certain kinds of modifications known as acetylations, that can help turn on DNA transcription, so actually reading the DNA by opening up the chromatin. But if we have modifications, particularly methylation, that can lead to more tightly packing or more tight packing of the chromatin, leading to condensation and turning off. So if you think about here, I'm a little RNA polymerase. I'm trying to kind of fit in these spaces. That works a little bit better than here, where I'm a big kind of protein. I'm not able to kind of fit in there and do the work that I need to do. All right, so what can we actually do to see what's going on, right? It's great to talk about this stuff, but obviously we can't see with the naked eye. It's a little bit more challenging to actually do those experiments. So there's a couple experimental assays that you may have heard of or worked with before. So first is ChIP-Seq, or chromatin immunoprecipitation sequencing, which is going to involve precipitating those protein DNA complexes and sequencing those chunks. So this is going to allow us to map the distribution of histones and try to understand where they are in the genome. RNA-seq is also a really important part of epigenetic work. So RNA sequencing helps us understand the genes that are being expressed in a given environment or a given time. And so if that chromatin is tightly packed, we're not going to be able to transcribe any genes there. So that might indicate that there's some kind of epigenetic protection of that area going on. So it can kind of help us assess what kind of epigenetic changes are happening and where. ATAC-seq is kind of relatively new approach, but this is going to actually identify open chromatin regions, which are accessible to transcription and other regulatory proteins that might be involved in maintaining that chromatin structure. And finally, an approach, and there are others in addition to this, but thinking about additional validations that can be done, you can use CRISPR to validate causal relationships between epigenetic modifications and gene expression. So if you cut out a specific region of the genome that's responsible for binding and creating that epigenetic structure, you delete it with CRISPR-Cas9, or you change it and see a different change in structure, then you can kind of show a causal relationship there. And so we're going to talk mostly about ChIP-Seq and ChIP-Seq results today, but just want to give us an idea of what this might look like. I mentioned that ChIP-Seq looks for that DNA protein complex and tries to detect, how abundant is that? Where is it in the genome? And so here's an example from a paper from a couple years ago where they're looking at signals of a protein called ChIP-Seq, and it's responsible for some of that chromatin structure. And so what they were interested in is understanding if that was consistent across different cell types. And so what they see is this particular region in the genome that I've highlighted in this green box, they kind of consistently have this signal. And so that indicates that there's consistently some chromatin structure happening there and some epigenetic modification, even if the sequence of the DNA itself is not different. Okay, so that's a little bit of the background there, but let's actually get into an experiment and try to see some of these results live and in action on AnVIL. And so we're going to visualize some results in a format called narrow-peak. You might see these online. Often they're published alongside the raw FASTQ files and things like that. So let's play around with that a little. But first, just a little bit more background of this particular study. So I mentioned the ChIP-Seq protein, which helps shape chromatin structure. And we're going to be focusing on the results of a study, Corey et al. 2020. And what they found was actually really cool is that ChIP-Seq attachment on places in the genome is not consistent. So they find that certain sites are especially resistant if we're trying to get rid of that ChIP-Seq protein. Some sites are especially resistant to depletion. So they hold on super tight versus other sites may be a little bit more flexible in terms of how strongly bound that ChIP-Seq is. And so these persistent sites, what they think are that these are key structural regions of DNA. It's really problematic if they get changed or knocked out. So we're finding these persistent sites are just really, really important and tend to stick that way versus being able to be removed. Okay, so here's the study. If you're interested, it's kind of cool. You can check it out here. But in their study, they're focusing on a specific locus. It's the Callicrane locus of chromosome 19, which is actually really interesting. It's got this series of these Callicrane loci all the way from 1 to, I think, 14. And some of these are really important biomarkers for cancer, and particularly KLK3, or also known as prostate-specific antigen, or PSA, is really important for understanding and detecting prostate cancer. And so if we take a look at this diagram they've got here, the ChIP-Seq protein is in this blue bar. And in its normal state, we can access some of these genes and transcribe them here in green. But you can see these ChIP-Seq proteins are kind of attracted to each other. They're bound up, and they've made this little loop here of KLK5 through 14 that they're not able to be accessed. So these proteins, this epigenetic modification, is actually protecting these genes and keeping them turned off. But if I were to take CRISPR and actually delete this area where those proteins attach, I could actually open that up to transcription. You can see they're in green now. They're able to be turned on. So these are, this particular locus is what we're going to be looking at. And it's, we can see kind of this binding here is pretty important for keeping these genes turned off. Okay, so let's jump in. There's a couple things we'll need to keep in mind before we actually jump into the activity. First is this GeoID. So we want to actually get the results of this experiment and bring them onto AnVIL. So we got to know what experiment ID that was. And a little bit of information from the manuscript itself. This Cali crane locus. I don't like study it in my day-to-day life and have the area of the chromosome memorized. So I wanted to get roughly what areas of the genome it's in. Okay. All right. So let's take a look at some stuff on AnVIL. All right, so I am in anvil.terra.bio, and I'm in a workspace that I've created kind of just to look at this work here. And I've got an analysis notebook here. So this is really similar format to what you may recognize if you work with R in your day-to-day life. It's an R Markdown notebook. You can also have R script files in your AnVIL workspaces. And so I've been working on this project a little bit, and I've already started a cloud environment, but it's paused for me. So I'm going to go ahead and resume that. Might take a minute or two. But I've set up this this workspace to do specifically this analysis, but I don't actually have any of the data on here already for this particular notebook. I'm actually going to bring the data in from Geo and work with it there. So this will take just a second to resume. But again, we're going to look at this particular notebook. And the nice thing is, if you are playing around with things on AnVIL and you don't know that you necessarily want to launch a cloud environment yet, you can take a little bit of a preview, read only, and take a look at the notebook, and you can see it interprets it. So if it's a notebook that has markdown elements, it'll print the title and the headers and the text and such, and the code will be formatted a little bit differently. All right. So let's go ahead and open that up. All right. So we'll need a couple things to get started. So first things, we'll need to install and load some packages. I'm going to go ahead and hit this because it takes just a second to load those libraries up. If you're launching this in a brand new, let's say you cloned the workspace and you made a brand new RStudio bioconductor environment, you might need to install some of these. So that code is right here. But we're going to need a couple of things to do some visualization of these results. First thing is our bioconductor package manager. Luckily, this is already installed on AnVIL, so we don't have to do that. But as well as the tidyverse, so we don't need to install that one either. But there's a couple that we'll want to install. So GeoQuery is going to help us download existing data from Geo, the gene expression omnibus, these results from this study. Track Viewer is really nice, handy package for taking a quick look at peak data. So if you have peaks from areas across the genome, you can kind of visualize them similar to that example that I showed earlier. And then we'll want a couple of packages from Bioconductor to do some human gene annotation. Okay, and so we're loading those up here so they're ready to go. You can see some of the messages from attaching those packages. I'll go ahead and collapse that down. So first things first, we have our Geo ID here, and we want to actually contact Geo and download some of those files. And then those files are usually compressed. We'll want to unpack those and get them ready to use. So let's go ahead and run this. We'll see those files populate here in my files pane. The cool thing is I don't ever have to touch any of these files, right? So if I'm working on a tablet, I can do this. If I'm working on my smartphone, I can do this. Basically, I don't need file storage on my machine. I can directly access those files from Geo. So this Geo query package is so cool. Very, very excited about it. Love to use it. All right, so we can see a couple of files have downloaded here. It's more than we need, but we're really interested in these narrow peak files. Okay. So the nice thing about these being on Geo too is that there's some metadata. I'm not just downloading a raw file attached to a paper. There's actually some requirements for experimental information. And I can do that here. So using the get geo function, I'll go ahead and get the series file information and metadata. And it's found two files. So let's pull out the data for both and figure out which we're more interested in. So this first one, let's get a little bit of information about the data processing steps. So I'll just take the first entry here for that series. So the first file, the first series that it finds is Affymetrix expression array. So this is a little less what I'm interested in. It's definitely relevant, but it's not the ChIP-Seq data. So let's take a look at the second one. Okay, so a little bit of information. They used Bowtie, a pretty well-known aligner to align ChIP-Seq reads. So I think I'm more interested in this one and visualizing some of the results. So let's take a look at some of that metadata. I'm just going to select that and take a look at the title and some of the characteristics. Okay, so in this particular data file or data series, I've got a bunch of different cell types. They have this RNA silencing. So they're trying to deplete this CTCF binding ability. And then I have some controls as well. But we can kind of see, okay, some of these are related to CTCF. And more information about their treatment. So non-target versus CTCF. So maybe comparing those would be pretty useful. Okay. All right. So here's where that location of interest comes in handy. I may not want to look at the entire genome, but actually just this chromosome 19, this Cali-Crane locus, because we're interested in seeing if any of those CTCF sites are particularly persistent, right? They're very important for structure. So I'll set up the range. And then I'm going to bring in those files that I just downloaded from Geo, and I'm going to read them in as the narrow peak type that I can then go ahead and visualize. So go ahead and run that to bring it in. And then maybe we want the names of those scores, those peaks, to be a little bit more descriptive. So I'm just going to set them to the name of the title here. And then take a look at those. Okay, so I have these specific cell types, these LN cap. These are related to prostate cancer cell line. The IMR90, these are lung fibroblasts. And so maybe I might be interested in kind of seeing if it's consistent across cell types. I've got control conditions, and then I have CTCF depletion conditions. Okay, so using that track viewer package from Bioconductor. I think is really handy. So let's just go ahead and run that and see what that looks like and if that plot is going to work for us. And this is, again, it's just the kind of default out of the gate plot. And it's not super easy to read, right? All these axes are kind of scrunched up here. And I'm not sure which sample is which, but I'm seeing some peaks, which is a good sign. So let's actually style this up a little bit. So I'll collapse that down. So if I take a look at these file names, you can see 1, 2, and 5 and 6 are control. So let me set those in this blue color. And let me take the depleted samples. So that's 3, 4, and 7 and 8. These low cell lines are really quite a bit different. It's a different experimental approach. So let's just not look at those for now. And let's rerun this with a little bit of color added. Okay. So a little bit easier to read. So I know the blue ones are the control, and then the red ones are the depleted version. And so what I'm seeing is some pretty consistent, I mean, there's a little bit of a gap here, but some pretty consistent peaks right here in the middle. And then some pretty consistent peaks up here on this far right side. But for the red samples, these peaks here in the middle, these are pretty low here. And then here, they're almost completely gone. There's a little bit of one right there. But these ones on the outside seem to be much more present. Okay. And so where is this actually in the genome? Let's add a little bit of annotation because this, I know it's the specific region that I'm looking at, but maybe others don't. So let's add a little bit of annotation so we can see what genes these are touching. Okay, so this is adding some annotation from the HG19 version of the human genome, which is what they used in this study. So we want to try to be consistent. Okay, so obviously this is pretty microscopic. It's a little hard to read, so maybe we want to zoom in on that a little bit. But I can see that there's some genes involved here. But the interesting thing is this set of peaks here is in a space. There's not a gene sequence right there, so that might be an important spacing region. All right, so let's clean things up. Obviously, like this is not looking super great over here on the left-hand side, and I might want to zoom in a little bit on these genes. So let's do a final bit of cleanup, run this chunk. All right, and let's pop it out, take a quick look at it. Okay, font's still a little bit small, but we could always adjust that later. All right, so I did mention the Kallikrein set of loci. We've got KLK3, which we know is really important for cancer detection. But really cool, we're seeing these results validated that we saw in that schematic. So this particular site here is between the KLK, I want to say KLK 4 and 5. And this set of peaks is attaching to this set of peaks here on the far right. And these are those persistent sites that Corey et al. Were talking about. These seem to be really important for structure versus these CTCF sites and peaks here in the middle don't seem to be as important. They are the first that are washed away when we do that silencing step. Okay. And the cool thing is this seems to be consistent across cell types. So regardless of whether it's those lung fibroblasts here on the top 4 lines versus the prostate cancer cell lines here on the bottom. All right. So kind of cool to be able to take their results in, validate those, and not have to do a ton of work on our own part to download and kind of work on those on our computer. Okay. So let's go back to some of these slides. So I based some of this on the vignette for TrackViewer. This is a really handy package if you're going to visualize epigenetic data or ChIP-Seq results. I think definitely worth checking out. And potentially be designing your own experiment and doing it at some point. So I just want to briefly touch on a few other AnVIL tools that you might want to use to do some of this analysis. Oh, thanks, Natalie. So option one would be to automate this stuff, right? So presumably we'd start by aligning our sequences to a human genome or even another species if that's what we're interested in, and taking that step through all of our samples. But there's multiple steps. And so our first option might be to check out the workflows on DocStore. So what I've done here, and I won't go too deep into it, but you can search terms like ChIP-Seq and find some of these pipelines already ready to go. If I were to click on this on the site and click through, there's an option to launch that on AnVIL to bring it back, basically. And so that might take a little finagling depending on kind of the attributes and setup of your experiment. But a lot of these are already set up in place. But if you're like me and you want to get into the weeds, there's always the option to DIY. So there's tons of excellent material from Bioconductor to get into ChIP-Seq and ChIP-Seq analysis. Particularly these two top ones are more like courses, so going through every step and helping you work through. These second two are more like vignettes for packages, so they have their own toy data, but you could sub in your data and work through it as well. If you are not so savvy on the coding and you would like to do point and click, I think Galaxy is an excellent option. So there is a really fantastic tutorial from the Galaxy folks themselves that you can run on AnVIL. So that's taking through every step of the ChIP-Seq through from alignment all the way to peak detection, and then some additional resources from folks outside of the Galaxy community, but these are also kind of nice. Okay, so one of the reasons we might want to use Galaxy and kind of redo this analysis ourselves is because this particular study, Corey et al., that I just talked about, they use the HG19 reference genome. And so what you might want to do is say, okay, well, maybe I want to take a look at some of these data, but I want to align them to the new T2T consortium reference genome. So the nice thing is we're able to do that. If I were to maybe take some of these FASTQ files and bring them into Galaxy on AnVIL, I could select that T2T consortium genome right here and simply kick that off to start that alignment. Okay, and then finally, wanted to touch on a really excellent resource that IFOMA has been really pivotal in putting together, and that is really just super introductory work in epigenetics using the UCSC genome browser. And so we have this activity here that you can check out. I'll go ahead and open that real quick. And it is going to go through some background material, just really introducing the fundamentals of epigenetics. And this is my favorite part, is if, you don't want to read through some slides or kind of watch a lecture, you can listen to some fun podcasts about epigenetics and some news articles and stuff like that. And then finally, there is a step-by-step exploration where it really tells you every step of the way how to click through the UCSC genome browser and look at those histone marks to understand how it is going to be able to turn expression off. So I think this is a great resource that IFOMA has been leading, that you could maybe use to teach others how to do epigenetics, or if you need to review, I think could be really helpful. Okay. So I know we're getting close to time here, or maybe over time a little bit. So just a couple things to wrap up. First off is we really appreciate your feedback. You can use this link. I'll try to go ahead and copy it here. Drop it in. Just let us know how this went and if it was helpful, what you'd like to see in the future. You can learn AnVIL in 2 minutes, believe it or not. So we have a series, a playlist where you can learn about scalability, for example, or sharing your data. Just a quick video to watch, while you're waiting for that next meeting or something really quick to fill the time can be really handy. And definitely keep in touch. Make sure you visit help.anvilproject.org, where you can keep the conversation going and let us know how things are working. If things are broken, we want to know about it. And then finally, AnVIL is much bigger than just these tools. So it's a data repository. It helps you build community and do all sorts of compute. So encourage you to check out those other resources. And many thanks to everyone who's been involved in this, particularly Afoma. I know she's just an epigenetics rock star. But yeah, this is definitely a team effort. So thanks, everybody. And happy to field any questions, and I'll stop share and hopefully we can have a little chat.